Welcome back to the show. I am your host, Ben McLeod, founder of Convey Bio. I'm here with Jesse McCool, the CSO of Bionova. Jesse, welcome to the show.
Thank you very much for having me.
Yes, so I guess we can just jump into it. So, recently you've made a move from Wheeler Bio to Bionova. Can you tell me a little bit about what your background is and what kind of prompted this change?
Sure. Yes, I'm a PhD scientist by training. I've been in CDMO for about 23 years. And Wheeler was my own baby. I co-founded it and got it started and handed it off nicely. It was laser focused on antibodies. My background is antibodies, proteins, a lot of biologics. And over the years, really looked for opportunities to get involved in advanced therapies. And so my rationale for leaving Wheeler and going to Bionova was an opportunity to build out advanced therapies capabilities for Bionova.
Right on. Excellent. So obviously one of the main things that you guys are working on is Plasmid DNA. Can you tell me a little bit more about kind of your unique process and what the tools are that you're using?
Sure thing. Yeah, we're building out a brownfield facility right now. It's about 10,000 square feet. It's a high titer plasmid DNA process. It's really turnkey. It's designed to be turnkey, really geared towards early stage companies. But because we have this line of sight to commercial, we're building out another facility, which we announced in June, building out about a 70,000 square foot facility for commercial manufacturing. We have the ability to support customers all the way from preclinical stages to commercial. So capabilities, it's 50 liter scale, it's all single use platform, high titer plasmid, standard workflow, standard process. Everything is on site from process development to analytical development, warehousing, QC, and then manufacturing.
Awesome. So just, I guess, diving into that a little bit more in detail. So right now the main goal is plasmids. Are there any plans to expand the offering to some of maybe the downstream products of plasmids? Yes,
absolutely. So we really believe that we can have an impact on the needs of customers by integrating plasmids with vectors. So we do have plans to add viral vector as well. Our first sort of step towards that journey is partnering with a technology provider in the US that is developing a next generation lenti technology. It's actually a 5 plasmid system, and so we are working with them. We're going to be their exclusive manufacturing partner for their technology. And really what it does is sort of upgrades all sort of Gen 3, Gen 4 lenti. And so we're going to be manufacturing that as well in the future. So we're looking forward to integrating that as soon as we bring plasmids online. We've done a lot of work on our clean room design. Really designed it to be purpose-built so that we can do multi-modality. We have a flexible operation in terms of the clean room design. And so that's our current building, and then our next building will be equally as flexible.
Excellent. Wow, a 5 plasmid lenti.
Yes.
That's cool.
More plasmids.
More plasmids. So I don't know how much you can say about that, but yeah, I'm going to have to look into that a little bit more.
Yep.
It seems interesting.
Well, the technology works with producer cell lines as well. So think about it as just an additional plasmid that really increases the infectivity of the lenti. So it creates a higher value particle and allows you to reduce your costs. As a CAR-T drug developer, you can reduce your vector costs significantly.
Interesting, very interesting. So I guess for right now, you're starting with research use only grade, but then a plan to also have GMP available. GMP lead times are pretty long, so this is probably something that's going to be exciting to a lot of people in the industry to really get this a bit closer to not real time, but within a reasonable amount of time. So when will that capacity be coming online?
Yeah, so the research use only is now. So we're making research use only plasmid today at typically 10 milligram batch sizes. The GMP grade is coming online later in the spring. So we're just in the final stages of there's a little bit of construction left, and then we go into EMPQ, and then we're ready to go. So sometime in late March, we're going to be making some announcements to let everybody know where we are with that. Again, we do have our headquarters being in Fremont, 10-year-old operation. We have a very mature quality system, so we're just tethered to that quality system. And so we've had to update the quality system just a little bit to accommodate the Houston's facility, but we're very ready to go and excited to make that announcement.
Excellent. So we started off this ATW week with a great panel with investors looking at kind of the growth of the space, around 25% CAGR. It's pretty exciting. Yeah. So I guess in terms of the field growing significantly, what would be Bionova's long-term plan to service the industry?
Yeah, it's hard to predict the future, but I do feel the energy there. I read a lot of the market intel. I do see the positive outcomes that are being predicted as far as growth goes. I think Bionova's mission is really to support the developers and really try to help them with solutions that are cost effective and agile and responsive, and that's really what we're designed for. So the Plasmid DNA operation being flexible enough to also accommodate vector, being able to compress timelines, really that's how we see we can add value. I think the main thing that drives Plasmid DNA is going to be the AAV and the CAR T markets. I think some things that will happen to really change is if we can solve for the solid tumor problem with CAR T therapy. I think we're going to see a massive shortage in manufacturing capacity to support all the supply chain components for that. And I think we're on the edge of that and seeing some successes on the solid tumor side. So there's a lot of manufacturing challenges to solve constantly, because this industry is constantly innovating, and but we're trying to be as agile as possible so we can be future proof and just continue to be a good partner for the industry.
Gotcha. So in terms of your outlook on the cell and gene therapeutics market, I'd be curious, particularly in the context of plasmids, what role do you think they're going to have in the future? Obviously, there's different producer cell lines that are starting to come out and stuff like that. Where do you see plasmids playing a role in the future?
Yeah, I think industry's very conservative, and so I think the 4 plasmid transfection system, for example, for Lenti, it's going to be around for quite some time. The 3 plasmid system for AAV is going to be around for some time. But yes, there are innovations out there with the producer cell lines that are going to reduce the need for the plasmid supply chain, and that's actually good for the industry because that's going to lower the cost. But these changes take time. As more progress is made in the clinic and more safety to humans is understood, I think you're going to see more and more ways like that to reduce the cost. And plasmid is a very big part of the cost of goods, and so I think that will continue to be driven down. But I don't see that there's any way we can continue on without plasmids in the future. Plasmids are going to be very important for gene therapy.
Yeah, for sure. So I guess just in conclusion, for customers who are working with different CDMOs, what do you think it's important for them to focus on when it comes to picking the CDMO that's right for them, when it comes to kind of avoiding maybe some pitfalls that you've seen in the past?
Yeah, I think it's a lot of CDMO selection decisions are made on the basis of relationships. I think relationships are very important. These are really important partners for you as a developer. You have to be able to pick up the phone and talk to folks that can make things happen, because science can go sideways, schedules can go sideways. So it's about relationship. I think that's a big decision factor. Another is it's quality, and so quality and being able to deliver enough plasmid per batch. So having a high quality, high yielding process is very important. So I think as we kind of emerge into the marketplace, I think we're going to be able to be very competitive on the quality and sort of the consistency and the reliability of our service.
Right on. Excellent. Well, this has been great. Thank you so much, Jesse. I guess maybe one other question that's kind of popped into my brain now, since we've got a little bit of time, would be, what do you think the future, in terms of what the industry looks like in 2030 versus right now, what do you think are going to be some key things to look out for?
I think we need to see continued success in the clinic for cell and gene therapy to continue to attract investment.
Yep.
Need to see some great results in the clinic. That is absolutely imperative for sure. We're all hoping this will happen because these life-saving therapies are incredibly important to families and patients, and it's been proven that they work in numerous cases. So there's some blockbusters coming out now, and I think that's going to continue to drive. I think we just need to see a few more clinical successes, and then I think the VC dollars will start to flow again. LPs will replenish that on the back end, and we'll continue to see a good trajectory.
Appreciate your insight. Excellent. Well, again, thank you, Jesse, so much for being on.
Thank you.
Thank you for everyone watching at home. We'll see you later.